Is working. Thank you for organizing this very nice satellite. So I'll be talking today about my PhD work that I did at ENS Paris. And I've been interested mainly in cis-regulatory motif and cis-regulatory modules. Well, again, another introduction, but very quickly, just that transcription factors, they bind these very small, somehow, binding sites of around 10 base pair, and they can combine on CRMs for cis-regulatory modules that interact with the transcriptional machinery to enhance or repress gene transcription. Just a very simple example of that, here is a limb enhancer that has a very specific pattern of expression in the limb buds in the developing mouse embryo. It's quite far from its target gene, so this is like a typical feature as well of the enhancers. And it's important in the development, so if you don't have this limb enhancer, the resulting mouse have basically no limbs, and in human, some just small variants in the sequence of this enhancer leads to very important developmental defects, here like polydactyly. So it's important to be able to locate these CRMs and to know what binds to them. And the usual pipeline to do that is to use models, so some simple binding models for a bunch of transcription factors of interest, scan the genome, and then use a combination of clustering of transcription factor binding sites, evolutionary constraints, so conservation in other species, some experimental evidence such as ChIP-seq data, and then predict CRM based on these features. And here I'm going to basically focus on two limitations on this approach, which is first, the limitation about the kind of model that you use, so the model accuracy in the beginning in the prediction of binding sites. And I will then talk about model exhaustivity, the fact that here I showed you using known models, but we don't know all models, and there is a need for de novo approaches. So this is basically the two parts of my talk. First, I'll be talking about an improved model of transcription factor binding. And second, I'll be talking about how to use knowledge of regulatory regions to decipher, to get the de novo motif from that. Okay, so let's begin with this first part. And the idea here is that we begin with ChIP-seq data. So we have a around 300 base pair resolution on the binding of a given transcription factor. And what we want to do is that we want first to use some simple model to get the candidate sites in this ChIP-seq data. And then out of that, we want to build more complex models of the transcription factor binding and use this model to actually find the good, maybe better binding sites that may or may not be different. The models that we have studied in this work, so basically the PWM models that you all know, where you suppose that the transcription factor binds the different nucleotides in an independent manner. Basically, the energy is just the sum over single nucleotide energies. And the natural extension of this kind of model is to add some interaction between nucleotides. So you basically suppose that when the transcription factor binds to DNA, it constrains pairs of nucleotides to adopt certain configurations. And in this kind of models that we named the pairwise interaction model, or PIM, you can have much more parameters than the simple PWM model. And I will explain how we take care of not overfitting these parameters. And the other kind of model that we also studied is called a mixture model, where we suppose that a given transcription factor can have several modes of binding, and in each mode of binding, it binds according to a PWM. And this way, you can implicitly have a higher order correlation than just the second order correlations that are here. So we wanted to compare those kind of models. Just to give the simple example to get our mind clear, let's say we observe AA, TT only. Okay, so we have half AA, half TT. Now, if I use PWM, I will have half AT times half AT, so I will say I've got 1/4 of each possibility. Now, if I use the PIM or the mixture model, I will include these features in the model so that I actually have the good probabilities, and this can be measured by a log likelihood, for example. So this is how you can compare the models on the basis on how they are able to fit the observed frequencies. And the workflow that we use is that we have a bunch of chipseq for 27 transcription factors, both in Drosophila and in mammals, and they cover several transcription factor families, such as SOX, E2F, BHLH. So we begin with some initial PWM that is known for this transcription factor. We scan their chipseq data, we get the sites. Then we begin from a PWM, and we add one interaction at a time. Each time we add an interaction, we fit the resulting model by maximizing the likelihood of observing the data given the model. And we do that until we minimize the BIC, which is a criterion that allows you to avoid overfitting of the data. So we add just the right number of interactions that prevent ourselves to overfit the data. And for the mixture model, we do the same thing. We begin from one PWM, then we add one PWM at a time, fitting by maximizing likelihood, and in the end, selecting the good number of parameters by minimizing the BIC. Once we have the good models with the good number of parameters, we loop, we scan again the sequences for binding sites, have new binding site, do that again until we converge to a stable set of sites with a corresponding model. So this way we have PWM, PIM, and mixture model. So let's see an example with a twist transcription factor in Drosophila. Here are the 10 most represented sites in the chipseq data. In gray is their frequency. And in blue, you can see the frequency as predicted by the PWM, which does not reproduce what we see experimentally. But either mixture or PIM model are much better in actually representing these frequencies. Which is true overall when you look at all the binding sites. So this is a log-log plot of the model probability versus observed frequency in the data set. And you want to be along the diagonal, and you see that at least the PIM, so the pairwise model in red, is better than and the mixture model are better than the PWM, which fails at actually representing those frequencies right. This is true for other factors, so this is just other examples for estrogen receptor or for MyoD, where you can see that the PIM model gives a much better fit of these in vivo binding sites. So we've looked, basically you can have a big difference between the sites that are predicted between the PWM model and the PIM model, so which is quite important if you want to make predictions in your chipseq data. So now, actually, with the PIM, the nice thing is that we actually include explicit interactions, so we can look at where these interactions are. And what we saw is that if we look at the typical distance between interacting nucleotides, they're mostly restricted to nearest neighbor interactions. At least in mammals, you have like around 60% of the interaction between nearest neighbor, but it's not 100%. You've got like also other interactions that are at greater distance. And in terms of the position in the binding sites, the interacting nucleotides are mainly in the flanking regions of the binding sites. So in blue, you see the average site interaction, and so you can see it's in the flanking regions where actually the information contained in the PWM is lower. So this is basically you add information where the PWM didn't give you information. And it's consistent with what has been already seen in vitro previously, but this case is for in vivo binding. So this is the first part. Basically, this new model PIM allows to improve, compared to the PWM, the prediction of binding sites in vivo. You can have substantial difference in predictions, and we observe nearest neighbor interactions, mainly in the flanking regions of the binding sites, around the core of around like maybe 6 base pair. And the source code is available on GitHub, so please use it and compare it to whatever method you like. Okay, and this is published in PLOS ONE recently. And so the second part I want to talk about is a different point of view. This time you don't know the transcription factors, you don't know the models. What you know are regulatory regions, CRMs, that have a similar output. For example, these CRMs all have an activity in the neural tube at maybe E10 in the mouse. All right? And what we want to do is we want to first find the common co-regulators of these CRMs, and using these common co-regulators, we want to find CRMs with a similar activity in the genome. And so we have developed to do that IMOGene, which is an algorithm that is motif-based. So basically what it does is that it takes as input a bunch of CRMs, usually around 15 or 20, because it's data that is hard to get, because you know that they have a similar pattern of activity. We increase the size of the training set by adding orthologs in other species. Then we have a motif builder steps that I won't describe here, but if you have questions, or I can show you how we do, that is Bayesian, and that takes into account an evolutionary model to build motif out of this bunch of sequences. So we end up with a list of motif, of PWMs. We rank them by how much enriched the training set is in binding sites for these motifs compared to background. Then we use the few first motifs, like the 5 first motifs, to scan the genome and predict CRMs that have high clustering in binding site, in conserved binding sites for these motifs. So let me show you an example of how it works. So what we did is that we wanted to compare with some state-of-the-art method in this kind of unsupervised learning from CRM, which are motif-blind methods. And so we used a benchmark that was published some years ago in mammals, where you take some CRMs, some mammalian CRMs, you learn motif on these CRMs, and the goal is to find a left-out CRM with a similar activity that is in 10 kb background. And you do that for all your CRMs, and you compute the number of times you actually find your left-out CRM. This is the sensitivity of your method, and you do that with 10 different random backgrounds to have a range of sensitivities. And so, so these are 2 examples in limb and in neural tube CRMs, and in dark brown is IMOGene algorithm, and we were quite happy to find that we are either in the same range as these motif-blind methods, or even slightly better in half of the cases. And so, well, we were interested with that result because basically we perform as well, but we also have motifs to display, which can be very nice because then you can go and mutate those motifs. So let me show an example where we did that. And this is an application to the Drosophila differentiation of trichomes. So trichomes are hairs that grow on the Drosophila larva. You can see here, their differentiation is triggered by shaven baby. Here is a known PWM for this factor. And what we did is that we took 14 CRMs that were known to be involved in this in the regulation of this process. We learned motif. We found that the first motif that was basically found by IMOGene was shaven baby, so the same a motif for this transcription factor, but with more refined information content. And we found a de novo motif that couldn't be associated to a known motif. And so we did mutations in 3 instances of this motif in different CRMs and found that we could abolish expression of a reporter of the activity this way. And we also used those 2 motifs to predict CRMs genome-wide and combine with expression data and ChIP-seq data to predict CRMs, 10 were tested and 8 were validated in the sense that they reproduce the same pattern of expressions as the training set and were dependent on the presence of Cheven baby. An interesting thing as well is that these CRMs had very different grammars of binding sites, so they had exactly the same output. So different inputs yield different outputs. And yeah, we have a user interface, so people can use it on the pass-through website. The source code is also available if you want to hack it or anything. And you basically, in the user interface, you just give some coordinates of your regions of interest, and we output the motif, where they are on the reference sequences, where they are in the orthologs, and also we predict some CRMs. Yeah, so to summarize, we have these methods that it's very good for small number of training set of CRMs in the training set, generates de novo transcription factors and can predict functional CRMs, and you can use it on the web. And yeah, it's published in NAR and the application in genome biology here. Thank you, and I really thank my PhD advisor, Vincent Hakim at INS Paris, and all these people that have been crucial to these different algorithms and studies. Thank you.